Plant breeding is a cornerstone of modern agriculture, involving the intentional manipulation of plant genetics to create new varieties with enhanced characteristics. This scientific endeavor has been practiced for millennia, evolving from rudimentary selection methods to sophisticated techniques that leverage the power of genomics and biotechnology. Plant morphology refers to the physical structure and shape of plants. Over time, humans have selectively bred plants to enhance desirable traits like bigger fruits, more vibrant flowers, or sturdier stems. Artificial selection is the identification by humans of desirable traits in plants and animals, and the steps taken to enhance and perpetuate those traits in future generations. For example, cauliflower, broccoli, cabbage, kale, and kohlrabi bear little superficial resemblance to their wild mustard relatives. And yet, through many generations of artificial selection, these 5 distinct crops were intentionally evolved from a wild, weedy ancestor. In flowering plants, male and female reproductive structures can be found in the same individual plant. The organ of sexual reproduction is the flower. Male gametes are found in pollen grains and produced in the anthers of the flower. Female gametes are found in ovules and produced in the ovary of the flower.
Flower formation. The plant develops flowers, which contain the reproductive organs. Pollination. Pollen from the male part, anther, of one flower is transferred to the female part, stigma, of another flower. Fertilization. The pollen travels down the stigma to the ovary, where it meets the ovule, egg. They combine to form a fertilized egg, zygote. Seed development. The fertilized egg develops into a seed within the ovary. Fruit formation. The ovary often grows into a fruit, which protects the seeds. Seed dispersal. The seeds are spread away from the parent plant through various methods like wind, water, animals, or even explosive mechanisms in the fruit. Germination. When conditions are right, the seed germinates and grows into a new plant.
Asexual reproduction produces individuals that are genetically identical to the parent plant. Roots such as corms, stem tubers, rhizomes, and stolon undergo vegetative reproduction. Some plants can produce seeds without fertilization via apomixis, where the ovule or ovary gives rise to new seeds.
Natural methods of asexual reproduction include strategies that plants have developed to self-propagate. Many plants, like ginger, onion, gladioli, and dahlia, continue to grow from buds that are present on the surface of the stem.
Let's discuss about difference between the old and the new methods of plant breeding. A method of selecting desirable individuals in a breeding scheme based on DNA molecular marker patterns instead of, or in addition to their trait values. A tool that can help plant breeders select more efficiently for desirable crop traits. MAS is not always advantageous, so careful analysis of the costs and benefits relative to conventional breeding methods is necessary. How genomic selection works. Collect data, grow a large group of plants and measure their traits, yield, disease resistance, etc. Build a model. Using this data, create a model that links specific DNA markers to desired traits. It's like finding patterns that predict which plants will be the best. GPSV. Predict performance for new plants. Analyze their DNA and use the model to predict how well they'll perform. This helps to choose the best ones to breed. Biotechnological vaccines in plant breeding, also known as plant molecular farming, involve using plants to produce vaccines and other medical substances through genetic modification. This cutting-edge approach integrates the fields of plant biotechnology and immunology to create cost-effective and scalable vaccine production systems. Example: The plant Nicotiana benthamina has been used to produce vaccines for diseases like COVID-19 and HIV. Gene editing in plant breeding refers to the precise modification of plant DNA to enhance desired traits or introduce new ones. This technique allows scientists to make specific changes to the plant genome without introducing foreign DNA, unlike traditional genetic modification. In summary, the integration of plant biotechnology and genetic engineering has transformed plant breeding into a more efficient and precise science, paving the way for innovative and sustainable agricultural practices. That's all from me. Bye and thank you for watching.